Okay. Can you sign the attendance? Where's the others? So this is classroom style. I don't like this classroom style. I prefer the discussion table. More like an exam. This is good lah to put your IC and then all your things. Ruler. Oh yeah, got ruler some more. Assignment. Okay. Mana yang lain? Ada class on the way. Okay. Because I want to talk on the assignment one first. At least we have a clear understanding on the assignment one. We start in 2 more minutes. Is that okay? Okay. So shall we start? Okay. So before we start, so I have a bit of announcement to make regarding the test. So I've uploaded. I've released the marks for the test. Quiz one I haven't released it yet, but the test is carry mark is 25%. So I've released this, it out already. So you may have a look at the marks that you have got in your test, uh, uh, and see whether you are satisfied with that marks. So second thing is the assignment one. So I haven't talked about this assignment one yet. So if you have received low marks in your test, don't be in despair, even though it's 35%. But the two assignments, we're going to help you in improving your carry marks. So do well in this assignment to help you in improve your carry marks. So this is the assignment one. So write a 3,000 word essay on the genetic disease. So I've asked before I think 2 weeks ago. I think that should be 3 weeks for our last class, right? So 1 week because we go to the industrial visit, and then another uh, week because of mid-semester break. So I think 2 weeks, right? 2 weeks. So I've at the end of the class, I've mentioned that book for the disease in the group. I think everyone have put in the list already. So you should have your essay on the comprehensive analysis, including the epidemiology of the disease. Especially, I would like to look at the genetic disease in comparison in Malaysia versus the worldwide. If that is more prevalent in Malaysia compared to the worldwide, or the vice versa. And what is the risk factors of the disease in Malaysia versus the worldwide, and the genotype and phenotype of the disease. So I think everyone know what's the genotype and also the phenotype of the genetic disease already. So describe what are those gene that is involved in this genetic disease. Okay, so this is the rubric of the assessment. So make sure that you have discussed each of these items in your essay. And I don't have the Turnitin, so do you want to have the Turnitin account set to you? I haven't set it yet. Okay, so maybe I send the turning in account for you. So send in your report in the turning in your essay. So I'm going to check your plagiarism of that report in the turning in, your format, grammar, cohesion and continuity, and the reference. So your reference should follow the reference style. So follow the APA reference style, American Psychology Association citation style. Okay, so I think that is for the assignment one. So the deadline is send it in. I think I give you another week. So send it before our class in next week. Is that okay? Because we have enough marks for the midterm BOE because we already have the test 35% before Friday next week. Okay. Besar kecil sangat. Okay. Tadi dia besar kan? Why suddenly become, oh, okay. So after I've looked into your test, so I just want to emphasize again on the previous lectures that we have, because I think some of you haven't get enough of the concept correct yet. So I'm going to go to the Mendelian inheritance first before we go to the non-Mendelian inheritance, because if you know about the Mendelian inheritance, actually that's going to help you in understanding the non-Mendelian inheritance. So that's why in your task, I try to cover all the Mendelian inheritance from the law of segregation and that second law of Mendel. So in the law of segregation, so we learn about the dominance. So from that monohybrid cross, so we know that only one form of the trait appears in the next generation. So we see that dominant, we're going to show the phenotype of that dominance, even though there's a recessive, so that's going to be masked off because of the dominant alleles. So in that work, in your working of that any of the inheritance calculation, please include the F2 family line as well. So don't stop at the F1, the offspring of that pure the breeding of that pure breed of that homozygous dominant and also the homozygous recessive. So some of you just stop at the F1. So in all of the inheritance working and also the calculation, please include the interbreeding of the offspring as well. Okay, so that's very important, because why we have the pure breed? Because from the pure breed we able to understand how the alleles go, because uh, pure breed will contain the homozygous dominant or homozygous recessive. Okay, so need to understand homozygous and heterozygous at this stage already, because it's already week 9 of the semester. So get to understand from your reading in the textbook what is the term of homozygous, heterozygous, and the earlier concept. Okay. So in this first Mendel experiment, the monohybrid crosses, so we understand it's always give you the ratio of 3 to 1. That is the interbreeding of the offspring that come from the purebred crosses. So the first generation after the crosses will always give you that 3 to 1. Okay, so some of you give me the first generation offspring only. You have to go in the second generation of that offspring to show that Mendelian law. Okay, so what is the dominant trait? So this is what we call certain alleles dominant, because it's more common than the recessive form. The dominant trait is stronger than the recessive form. A trait that is expressed if one allele for that trait is inherited. If this is question, right? So all of these are true if we have all these statements. So let's see. A trait is dominant will increase in that population while recessive traits decrease. So is that true or false? So it's false. It's always false within that ratio. So it depends on the genotype. So you not increase over time. So you're going to have that ratio of 3 to 1. So of course, when you have a lot of breeding, so you have a different numbers, but all this when it calculated statistically again of all this study, so you're going to give you the 3 to 1 ratio. Macam mana ni? The concept. Okay. So let's do more problem case study. Think you also can see from your test. So my design of question will be more looking at the working of and also calculation of most of this concept of inheritance. So we have Mendelian inheritance, and then later we're going to learn the non-Mendelian inheritance, such as the codominance, multiple alleles. Then you're going to be getting more confused. So let's move into this first. So if this person have freckles, so does the wife. Their son has no freckle. Is having freckles is dominant or recessive? So let's say it have a freckle and Derek have no freckles. So this is how making that tree. So no freckles. So make a family tree. So usually it's a recessive trait. So if it's recessive traits, then we can, if we have a certain genotype that we already know, then we can know what is the genotype uh, in his father or from the mother. Okay, so let's work on this. Pebbles have a smooth hairline, and then this person has a pointed hairline. But one of the parents has a smooth hairline. If Pebbles and Bam Bam will have kids, what are the odds that their child will have a pointed hairlines? So you have to recognize what is the dominant and also recessive. Then what's the possibility? Then you're able to get this. So if it's 50%, so that is because smooth hairline. I think this question, you should have the dominant, and this is not dominant, recessive. So I think we have stopped until here. Let me. So I didn't cover that in the last lectures. So law of segregation. So during the law of segregation, so this happened during the formation of that gametes. So these two factors were going to separate, and only one of the alleles ended in each of the sex cells. So in the homozygous parents, all gametes produced are similar, while in the heterozygous parent, two kind of gametes are produced in equal proportion. So the ratio compared to the law of segregation, compared to uh, the first uh, uh, law is the 9 to 3 to 3 and 1. So let's understand this, what's happening here. So in the homozygous parent, so we have the homologous uh, chromosome, and the gene or alleles will be at the homologous chromosome. So the gametes will have two identical form of the genes. So in heterozygous parents who have uh, the different uh, alleles in the homologous chromosomes, because we have pairs of these chromosomes in an individual, and this during the gametes, we're going to have two uh, homologous chromosomes with that alleles. Okay, so consider this genotype, a purple parents, a pure breed of purple and pure breed of white will have this. So the gametes of the parents will going to from sperm cells and the egg cells will have this offspring. So that is the first one. So that is the Punnett square. So that's going to give you that ratio 3:1. So this is still monohybrid. Okay, so let's move into this one. A person with heterozygous dominant A and recessive A genotype for the trait of A. So what allele for that trait that can be person put into their gametes? So it's A or either A in the small capital because of what we indicate is gametes in the law of segregation. So a flat feet are caused by recessive alleles. So if Bert and his wife Betsy have a normal feet, but their first child Blinky has a flat feet. So what are the genotype of Bert, Betsy, and Blinky? So first, you need to draw up. So we know that F is the recessive. So it means to have a flat feet. So you need to have a homozygous recessive. So we have the first child have, so we have Bert and Betsy. So then you have Binky. So in this, we already know the offspring of these marriages between the Bert and also Betsy. We're going to have a child that have a flat feet. So Bert and wife Betsy have a flat feet. So what are the genotype of Bert, Betsy, and Binky? So here we already know we have the F and also the F. So what do you think? Their parents. So means you have F. Small. Let's try it out. So we have F. So you have to put it into a Punnett square so that the resulting will get you um, the uh, the homozygous recessive. So we have FF. Okay. So what if we have a dominant? So let's try it out whether it can give the homozygous recessive or not. So here is going to be dominant, and then this heterozygous will also give the uh, what we call normal fit. This normal fit, this uh, normal fit as well, because you need to have the homozygous. So this can't be true. And Betsy and Bert is not having the flat fit, so that means it either homozygous, dominant, or heterozygous. So it can't be a FF in either two of them. So to solve them, you need to do the testing first, as what I did just now, in understanding in their offspring. So what going to be possible parents of the genotype of their parents. So what are the odds of Bert and Betsy next child having a flat feet? So this 3 to 1 already. So still the same probability is 1 to 4 probability. So the odds is still the same. It's not after having binky, so the chances is be different, because it's through the uh, segregation of these gametes, we're going to produce the next offspring. What is the chances? But if you have a lot of offspring, like what you can do with the peas, so you're going to reach to the ratio of 1 over 1 over 4 chances of getting the flat feet. So should we do this maybe later? Okay. So this, I haven't go through the law of independent reassortment. So I've stopped until here. So in the hybrid, it involves more alleles. So it based on the gene on the separate chromosome. Each pair of alleles assort into gametes independently. So this pair of alleles are passed to offspring independently of each other. So what we're going to learn today in the non-Mendelian laws, so there are certain other factors also. So here the assumption, when if the alleles segregate independently and you're going to uh, uh, be observed in their offspring. So if there is one or more than one trait, so we can do the dihybrid course. So some of these are using the peace experiment by the Mendel. So we can do the dihybrid course in fulfilling this the second law of Mendel. Okay, so what happened in these traits on a separate chromosome sort independently of one another during the gamete formation? So you can see the condition is the allele is in the different chromosomes. So if the allele is in the same chromosome, so this law doesn't apply. So look at the question carefully when you going to use this uh, 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 working models for your solving the question. So first you must understand the allele is carried on separate chromosomes, and this we're going to sort independently during the gamete formation. So during the replication, you're going to have all these alleles as we understand in that first law. When we have only one alleles only, so this is going to be independently assorted when you have that uh, uh, having the possible genotypes, and this will going to give you all the reassortment of all the possible alleles. So let's move into here. So in this Punnett square, so when the heterozygous from that pure breeding, so heterozygous that have these two traits that self-fertilize, so you can see all these possible fertilization, self-fertilization. So it's going to give you the ratio of 9 to 3 to 3 to 1. So when we have the dominant or dominant for the smooth, for this example, then you're going to have the smooth. So what happened in the homozygous recessive, same as previously, we're going to have it become a wrinkle seed. So the yellowish is the dominant. So when there is a recessive yellow gene, then you're going to be, the piece is going to be in green color. So when you calculate all this, it's going to give you the seed shape and the seed colors. We're going to have this phenotypic ratio. So in solving the dihybrid crosses, you need to begin with the parent's genotype. So assume that the two traits on the different chromosome. So pure breed can produce this gamete. So a pure breed of recessive can produce this gamete. So in the offspring of the pure breed of this homozygous for T and homozygous for dominant R, we're going to have all going to be TR in the capital letter, and the homozygous pure breed of recessive T and R, we're going to produce this, and all the resultant of the pure breed, we're going to give you the heterozygous of this T and R. So all of these offspring are tall and also purple flower. So that's the breeding of these tall and also the purple dominant alleles. So that's not all, but you need to, in the hybrid cross, you need to solve the offspring of the self-fertilization of this offspring. So you can have the possible assortment. So you need to make the possible the assortment. So in the heterozygous offspring, so we can have the possible of tall and dominant purple, and also tall and recessive going to give you the white color recessive, and also can have the recessive that going to give you short with the purple color, and short with the, so get all the possible gametes that can be produced, and then cross it as normally previously in the Punnett square. Then you're going to get this ratio. Okay, so let's solve this together. So if a person is heterozygous for dominant trait of A and B, so this is the genotype. So when we say heterozygous, then we know that is the genotype. So if you're not given this one, you can understand if heterozygous for trait A and B, so you already know that is the possible arrangement of the genotype. So what is the one possible combination of those alleles that can go in the person gametes? So you have AB. So this not possible. So gametes also. So back to our previous question just now. This is the possible in that organism or the itself. So this not possible. So it can be AB or AA. So it's only AB because we can't have these two together. So this is going to separate in the normal, the first law of segregation. But this is another alleles in the different chromosomes. So the possible is A and B. So this is going to be the homozygous dominant. Okay, so a pea plant with a purple flower and yellow seed is crossed with a pea plant with the white flower and green seed. So all the plant in next generation have purple flowers and yellow seed. If two plants from this hybrid generation are crossed, what will the offspring looks like? So let's do it. So try to do this. So a pea plant with purple and also yellow seed is crossed with this pea plant from with the white flower and green seed. So all will have the purple flower and yellow seed. So if all, usually if all have the purple and yellow seed, that means all are heterozygous. Purple and yellow. Why? So that means the parents, usually they are homozygous. Then only with that you're going to get all we're going to be the heterozygous, and this will going to give you the yellow, uh, purple flowers and yellow seed. So the question can ask you to determine from this heterozygous, determine what is their parents. So even though it didn't say the parents are homozygous or pure breed, we know that from this statement, all the plant in next generation have purple flower and yellow seed. So we already from that ratio, we understand their parents must be pure breed. So if the two plants from this hybrid generation are crossed, so what do their offspring looks like? Do a dihybrid square and predict the outcome. So do the working steps that I've mentioned just now, step by step, what is the Punnett square, and give in the ratio of the all the possible genotype out from it. Okay, I think this is the same thing. So maybe I let you to do this later by yourself. So this back to our today, this week lectures. So if these two Mendelian laws going to cover all the possible inheritance that we see normally. So since the Mendelian, so there are various instances or things that doesn't fall into the law of the Mendel, because we're going to discuss this later in our later chapter from what we have learned from the Mendel inheritance. So we have the codominance, multiple alleles, epistasis. So they're going to affect this ratio that we have discussed so far. So as we know, genes may have multiple alleles. So depending on where these alleles, some may have codominance, some may have incomplete dominance that we're going to see in the later chapters. Okay. So sometimes in the non-Mendelian inheritance, so that's what we're going to investigate and discuss here. So it depends on the gene interaction. So sometimes it's going to give you incomplete or partial dominant, codominant, and some multiple alleles of the genes. So certain genes may responsible for different traits, or one gene we're going uh, as what we have discussed before this, one gene responsible only for one trait. But here, one gene may be responsible for many of the other traits. So let's understand what's alleles again. So it's alternative form of a gene, one member of a pair that located at specific position on a specific chromosomes. So this is very important. So what you need to understand, you have pairs of chromosomes. So in your human body, so because different organisms have different sets of chromosomes, in your own body, you have 23 sets of pairs of chromosomes. So in total, the chromosomes will be 46 chromosome. Okay, so I want you to understand that. So alleles, one allele may be at a specific position on that specific chromosomes, one member of a pair. So this is what I mean by alleles. I think that's an example we understand by now. So in incomplete dominance, so here one allele is not completely dominant over the another alleles. So when we have a dominant, let's say dominant of red color, and then recessive going to be give you a white color flowers, so recessive. So when you have the heterozygous, so what do we expect in the incomplete dominance? So we know this is dominant gene. So when we have the dominant alleles with the recessive alleles, so usually we're going to give you red in the Mendelian inheritance concept. But in incomplete dominance, so the heterozygous will not turn to be red in color because it's just a partial, it's not able to fully affect on the traits that is red color. Here maybe it's going to be in the middle, maybe become pink in color. So it become like the intermediate from the red and also the white color. So still remember that I've mentioned that when a certain trait from your parents mixed with another trait that you see from your paternal and maternal have a different traits like uh, black color eyes and then have a green color eyes from your mother. So you don't have the mix between these alleles. But in incomplete dominance, so you may have the mix because these genes are not fully dominant. So it's only partial dominance. So you're going to give incomplete dominance effect on the offspring phenotype. So that is what mentioned here. So the resulting offspring are in the intermediate colors in between of their parents. So this, you can see it in the what we call 4 o'clock plants, or in bahasa Melayu bunga seroja. Have you see that before? Bunga seroja is always in the bushes. So some may have a red color, and then sometimes you see it is in a pink color. So sometimes in incomplete dominance, you see this doesn't follow the law of the Mendels. So we can describe it because it's based on the incomplete dominance. Because the offspring phenotype is intermediate between the phenotype of its parents. So what I've described here, so in the homozygous, a pure breed of homozygous red color uh, 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 4 o'clock flowers, and also the homozygous recessive of these 4 o'clock flowers, when it breed within each other, we're going to resulting in the heterozygous of uh, the red color. So this resulting incomplete dominance in pink when it interbreed. So we're going to have this ratio. So you're going to have this 1 to 2 to 1 ratio. So we have 2 pink color, 1 red color, and also 1 white color. So this is another example. So good example is to make so that we're not confused with the uh, Mendelian law. So usually we just put it as the color, and then we put it in the what we call superscript. So for this example, the red color is the dominant. I think I prefer to still put it, if I solving this question, I like to put this as the dominant one. So this, the pure breed, we're going to give you the red color, and then this will going to give you white color. So the resulting will give you the heterozygous, and then when this interbreed, you're going to have so we have CR and the color red, and C and CR. So when these two, we're going to give you white color, and then this going to give you red color, while here we're going to get you the intermediate. So when there is heterozygous, okay, then you're going to get you pink color. So this is because this dominant gene will going to have incomplete dominance over the recessive. So that's why it's going to give you intermediate. So if you're given the ratio of the offspring at these three different traits, so without telling you this is incomplete dominance, and you are given the ratio, so you know this is incomplete dominance law need to be applied to your working steps. Okay, so this is a third phenotype appears. So usually we know that only in the Mendelian inheritance, two phenotype always appears. So when there's third phenotype appears, so that must be something not following the Mendelian law. Okay, so this is the example. Okay, so another that is not following the Mendelian law is your blood type. So any one of you A type, me myself is A type, and then B type, and then AB. AB yang paling bagus lah. Can receive all the bloods from the other people. O yang paling bagus lah. They like to give blood to the everyone. But when they have they themselves have problem, then nobody can give to them. Okay, so let's discuss the example of the blood type. So here we call it codominance. Okay, so we have just now incomplete dominance. Now we have codominance. A good example of codominance that we see that deviate from the Mendel's law of inheritance is the codominance. That one example is your blood type. So the differences between the codominance then incomplete dominance. Okay. So for example, just now used back the pink flower may be the product of two alleles that produce red and white pigments that become mixed. So codominance on the pigment level, no dominance on the color levels. Or the result of one alleles that produce the usual amount of red pigment and another non-functional alleles that produce no pigment. So as to produce a dilute intermediate pink color, no dominance at either level. I can't remember this statement. So maybe we use the codominance in the in your blood type. So So why I choose the color when it's not non-Mendelian inheritance, I think is properly we do like this so that we can easily show this is non-Mendelian inheritance calculation. So why we use a certain alphabet on this? So you can refer to the genetics. So there are certain alphabet that the geneticists always use when we are referring to a certain phenotype. So for example here, they prefer to use I for the uh, red blood red blood cell. So in a blood group, the genotype of A, usually they have this alleles of A. So or without the uh, without the what we call that protein of A, then we're going to get the blood type A. So the possible uh, for blood type B is you have the both B or one B and one without the receptor. So if you are AB, then you have heterozygous for the A and B. So you can't have because it already filled up with another one. So for O, you don't have any of this receptor of A, either A or B. So in relation, for blood type A, you can receive blood from A or O, and if you are blood type B, you can receive blood from only person from blood B and O. And if you're AB, you can receive from anyone. And if you're O, you only can receive blood type only from O. So to make this more complicated, I don't want to discuss on the research. So you know what's research, positive or negative. So it's quite complex if I discuss it all together. So to make it easy, so I discuss on the receptor A and B and without the uh, alleles. Okay, so for codominance, also in the sickle cell disease as well. So in heterozygous condition, both alleles are expressed equally. So if you are in a normal without any sickle cell disease, so you are either have it in a normal alleles, both, or the heterozygous. So if you have the sickle cells alleles, when you have it both, then you're going to express equally. So if you have heard before the sickle cell anemia, that means you're going to have red blood cell that carries less oxygen because the cell is in the most of them are in the sickle cell, so you're going to be less oxygen in their body. So in the codominance, if that sickle cell, you're going to maybe uh, express equally, so you don't have any issue. But if it's all sickle cells, then you have issues on the level of oxygen being delivered. Okay, So to show the individual with sickle cell anemia, another who is carrier but is not sick, but because uh, if you have the sickle cell and the normal cell allele, so this will not give you a sick, but they are carrier. So when it have both of that sickle cell alleles go together, then all we're going to be RBC, we're going to be in the sickle shape. So this is to show um, it depending. So this is to show if the parents is one is a carrier and another parent is a sickle cell anemia patient. So you're going to have all their offspring. Uh, Half of them are sick and half of them are carrier. So it's always uh, advised if their parents know that they are carrier, so it's advisable not to marry a person that have the sickle cell patient. So if it's normal person but it's not carrier, marrying person that have the sickle cell, then most of them are going to be carrier, but they are not sick. So that is uh, that I would like to say from here. Okay, so let's discuss on the multiple alleles. So multiple alleles is when there are more than two alleles possibility for a gene. Okay, so there are more than two alleles for a trait. So for example, the blood type A, B, AB, and O. So the blood alleles uh, are these. So this example of multiple alleles, I think that's to explain what we have discussed just now. Okay, so if we have the allele of A, allele of B, and allele of the I, the recessive one, then it produces no antigen. Mm. So this is how the blood type A looks like. So blood type A, we're going to have this A antigen, and blood type B will have the B antigen, and blood type A and B we're going to have this both antigen in the blood cell. So this is how the phenotypically this blood type we're going to looks like in the cell. Okay, so let's cross between these multiple alleles. Think this what that we're going to expect if we know that the mother has a O type blood and the father have the AB blood. So we know that the father would have the A and also the B, and for O we will have the recessive that doesn't have any of those A or B. So we're going to have uh, the phenotypes as being mentioned here. So we have the A, the B, and then another ratio for A and another ratio for B. So we're going to have either its offspring is type A or type B blood, because uh, the parents have the O and also have the AB bloods. Okay, so in another scenario, if the mother is heterozygous for type B blood, so for type B, so you need to be homozygous for B or heterozygous. So if you are not, if you're given this statement, you already know that heterozygous for type B, you know you are given this genotype of the for this father, for this mother. So a father that is heterozygous for type A blood, so similar things here. So if you are given homozygous for A, so you know the genotype of the parents is this one. Okay. So if you do the crosses, so you expect to get this ratio. So you get the AB, the B, A, and O. So you get all these four ratio. Okay, so I think in our next class, I think I want to talk more on the X-linked traits, uh, and also going to give you talk about the epistasis. So maybe I give this link and then go and watch the videos on the X-linked traits, multiple alleles, and incomplete dominance, codominance, and epistasis. Not yet. I have the other slide. This is the slide from last semester. Okay, so this is slide that I prepared by myself. So we have talked about the alleles, incomplete, codominant, and multiple alleles of genes. Think, wait, oh, in the same slides. Okay, let's do one example. Think for the Mendelian and non-Mendelian genetics. If we learn a lot of example, I think that it's going to help quite a lot. Okay, where is it? Mm, okay, let me find a question. Okay, let's try this question. So a maternal effect gene, I think this is a X-linked gene. So I haven't discussed this yet. Okay, let's try this. So you have a female snail with a shell that call to the right, but you don't know what's genotype. Okay, so usually the question is always like that. It's not given for the certain parents genotype, but you can know and find it out from the other uh, 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 tips from the question. So you may assume that the right coiling D is dominant to the left coiling in the small d. You also have a male snail of known genotype. So how would you determine the genotype of this female snail? So what do you do? In your answer, describe your expected result depending on whether it's a female, it's D, the homozygous for the right coiling, heterozygous for the right coiling, or the recessive for the left coiling. So how do you determine the genotype of this female snail? So you have to utilize your knowledge on what we have learned so far in solve this question. So we do not know that female snail, so it's not given. So we do not know what's and then for the male snail of you know uh, of known genotype, but it's not given to you. How do you determine the genotype of this female snail? So we have DD, the heterozygous. So you must give assumption of this one. Mm. So this is not known. So you have females call to the right. Okay. So go to the right means it's either D. So that is so far what we know. So how that we know? Uh, Expected result. So it depends. So give you assumption whether your female, your male of that known either is DD or DD. Okay, so you must do these two one and then the one possible with all these possible crosses. Then uh, see whether, but this question, if we can improve, if this is given, that is much more easier. Then we know whether what is the uh, female genotype will be. But here you're going to have a lot of assumption. So here, how you determine the genotype of this female snail? Then you must make the Punnett square for all the possible uh, crosses, and then what's the possible resultant offspring. Okay, so this is how you answer it. So it's not direct because I think this question is quite high in cognitive level. So I've chosen a quite difficult one. And then describe the expected result. So you may have a different ratio based on the uh, resulting uh, assumption that you have made from this question. I think this question is difficult one because this is application and experimental question. Okay, let's try this. A Drosophila embryo dies during early embryogenesis due to a recessive alleles of maternal effect called bicort. The wild type alleles is designed by bicort. So what are the genotypes and phenotype of the embryo mother and maternal grandparents? So this is using the information that we know from the offspring. So we're going to work it backward to know what is the possible genotype and phenotype of the mothers and also the first generation. So this is the second generation. To know what is the first generation from the